Okay, this is going to be our final lecture, not just for this unit, but for the whole semester. So first, I just want to go ahead and say thank you so much for all of your hard work. This class is very intense. It is not an easy course. So I really appreciate all of the attention that you've given it and the work that you put into it. It shows, and thank you so much. All right, let's go ahead and finish up chapter 16. So we ended the last one with the structure of the eye. Now we're actually going to get into the physiology of vision and how sight works. So starting with refraction of light. Sharp vision requires light rays to be bent or refracted as they pass towards the retina. So refraction results when light passes between media of different densities, such as the air and the cornea, that outer structure of the eye, or through curved surfaces, such as the lens, the more internal structure. And every object has a refractive index that's going to show how much it's a comparison of density to show how much light or an object will be refracted or bent, such as a spoon, when light passes through it. Okay, so to see light properly, we have to be able to bend that light. Okay, so focusing has, there are four different methods that are used for focusing. The near response is for objects that are closer than 20 feet. Your eyes converge together, the lens accommodates, and the pupil constricts. Okay, so convergence of the lens is when your extrinsic extrinsic muscles pulls your eye medially. It directs the image of interest onto both foveas, and weak eye muscles may cause diplopia or double vision. So this is when you're trying to focus in on one thing, your eyes kind of come in together. If you're trying to look at the tip of your nose, your eyes cross, so that's convergence of your eyes. Accommodation of the lens, your ciliary muscles contract to thicken the lens. The slackened suspensory ligaments allows that lens to get a little bit less flattened, and refraction increases as the lens gets thicker. And your pupil constricts. The sphincter pupillae, remember there were the dilator pupillae and the sphincter pupillae muscles. The sphincter pupillae will contract to shrink the hole of the sphincter, or the pupil, and light passes only through the center of the lens, avoiding blurriness that would result if the thin edges of the lens were used. So it really kind of focuses that light in onto the retina and the fovea. For objects that are further than 20 feet away, your eyes are going to face forward. So even still, this is only, 2.5 feet or so from me, so my eyes are still fairly converged in. But if this were farther away, then my eyes would be more pointed forward. The lens is flattened, so as opposed to the ciliary muscles contracting, your ciliary muscles are going to relax, and the suspensory ligaments are going to pull taut, so that the lens flattens and it has less refraction. Light has less refraction. And the pupil is relatively dilated to allow a greater amount of light into the eye so that you can get a better picture. Okay, now getting into phototransduction, which is actually converting light to electrical signals. Something I've kind of hit on a few times is that in the nervous system, we're always usually converting some modality into electrical signals, whether it's a chemical signal into an electrical signal, in this case, a light into electrical signal. We'll see for hearing, we're conducting sound into electrical signals, vibrations into electrical signals. So this is a common thing for the neurological pathways that we have to convert to electrical signals. It's performed by photoreceptors, your rods and your cones, the photoreceptors that we have. And looking at their different parts. So photoreceptors have an outer segment that extends into the pigmented layer of the retina. Remember, the retina has its superficial pigmented and then the deeper neural layer. So the outer segment extends into the pigmented layer of the retina, and it has hundreds of photopigment containing discs that absorb light. These discs are going to be continuously replaced. You have your inner segment that contains the cell organelles. The cell body has the nucleus, and the synaptic terminals contain vesicles storing glutamate neurotransmitter. We'll see glutamate plays a role through the transduction of light. Right, looking at the rods and the cones. The rods are longer and narrower than cones, and they're more numerous. Each rod is highly sensitive. It's activated even by dim light. It's also noted that in bright light, rods tend to be less effective. So rods are really good effective in low dim lighting. The periphery of the retina is going to be where most of your rods are. So in lower lighting on the periphery of the retina, we're able to see more clearly. And many rods converge on fewer bipolar cells, which converge on fewer ganglion cells. So this results in sensitivity to dim light, but a blurry image. Essentially what it's saying is you have a lot of rods, they're sending their signal, and their signals are converging onto fewer and fewer ganglionic cells that actually send a signal to the brain. Your cones are concentrated at the fovea centralis. Remember, that's the area of sharpest vision. They're activated by high intensity light and allow for color vision. So while the rods are working in our low dim lighting, the cones are more activated during bright light and for color vision. Cones also have a one-to-one relationship with bipolar and ganglionic cells so that we get a sharper image, but only possible in bright light. They only work in bright light. Right here is a depiction of this. So you can see in this pigmented layer, you have your discs of your rods and your cones. And then you get into your neural layer, you have your inner segment, the cell body, and these synaptic terminals that are going to synapse with ganglionic cells. And just so you know, the light, it's showing this coming here, but light really is coming from, this is the outer segment, so it's coming down, and then this is going to be working towards the brain on this end. All right, photopigments. Photopigments are the light-absorbing molecules within the discs. They're found within the membranes of the outer segments of the rods and the cones, and they're made of opsin protein and a light-absorbing molecule called retinal, which is synthesized from vitamin A. Okay, so you have photopigments that are in the membranes of these rod and cones, and they have this opsin protein, which is just a basic protein structure, and then inside of that protein structure is going to be this retinal molecule that's going to be absorbing the light. Different pigments, different pigment types have different opsins, transducing different wavelengths of light. So basically it's saying is that you have, for each wavelength of light, you have a specific receptor that's going to respond to it. So each photoreceptor has only one photopigment type. The rods contain rhodopsin. That's their, so opsin protein. So in the rods, rhodopsin. And then the cones have what's called photopsin. And there are three different types of photopsin molecules in the cones. You have blue cones, green cones, and red cones. The blue cones detect short wavelength, green intermediate, and red detect long wavelengths best. So you have these light-absorbing molecules that when light hits them, they absorb that color, they transmit it depending on the color is absorbed rather by the specific photopigment, and then that signal can be transmitted down through ganglia. So here you go, you can see your blue cones, your rods, your green, and then your red cones, all absorbing and working in slightly different wavelengths of light. And again, they'll have their optimal wavelength. So you can see that it is a range. It's not like, you know, the green cones are only in this one particular peak. The green cones definitely do have a range of colors, even stretching into these yellows and reds, or red, yellows, and oranges, that they can possibly detect. So it really is going to just be, there's a little bit of I'll say, gray area here when it comes to how they absorb the color. You know, you're not going to have one specific cone is going to absorb this one specific wavelength. It's going to be able to absorb a range of wavelengths, but specific to that cone. Okay, so continue with phototransduction. In the dark, so when there is not any light stimulation, and especially in dim lighting, your rhodopsin contains cisretinal. As light starts to hit into the retina, that reconfigures it into transretinal. Having that light energy hit the cisretinal converts it to transretinal, which dissociates from the opsin protein, and this is called the bleaching reaction. Okay, so light hits, it converts cisretinal to transretinal, that then dissociates from the opsin. After this bleaching happens, rhodopsin must be rebuilt for the rod to function. So to be able to transmit light signals, it has to be put back together into this cisretinal form. And this is a very slow process. So once the rods are bleached, it does take, it can take, I think, up to 30 minutes sometimes for us to readjust back to the light. So the rods don't function well in bright lights. In cones, the process is similar for photopsins, but it's much quicker. It happens a lot faster. That rebuilding function happens faster, and so your photopsin regenerates much more rapidly and allows the cones to function during bright light or in bright light. Here is just kind of a breakdown of that. So light is absorbed by these rhodopsin molecules. Cis turns to transretinal. The transretinal dissociates from opsin as opsin becomes activated. When it says that opsin is activated, this means that it's going to allow for it to transmit a signal. Transretinal is reconverted to cisretinal in the dark, and then the cisretinal associates with opsin to reform rhodopsin and deactivate it. Okay, so it's activated to send signals in the light, deactivated to not send signals in the dark. Okay, so I have a YouTube video for us to watch. I'm not gonna lie, it's like 5 minutes long, but I think it does a really good job of explaining this because vision can get a little tricky. It can get kind of hard to explain on my end and to understand on your end.
depolarizing.
Okay.
The retina contains two types of photoreceptors, rods and cones. Both types have an outer segment composed of membranous discs containing light-sensitive photopigments. Each also has an inner segment that contains the cell nucleus and gives rise to synaptic terminals that contact bipolar and horizontal cells. Phototransduction is the process of a cell absorbing light and creating a response. The response is a change in the amount of transmitter that is being released onto target neurons. The photopigments lie within the disc membranes of the outer segments. In rods, the photopigment is called rhodopsin. The 7 transmembrane domains of the protein portion of the molecule, called opsin, traverse the membrane bilayer. The opsin forms a pocket in which the light-absorbing portion of the photopigment, a molecule called retinal, resides. The photopigments in the rods and the 3 types of cones differ in the protein portion of the molecule, which tunes retinal to absorb specific wavelengths of light. Here, retinal is in a conformation called 11-cis, with cis referring to 2 hydrogen atoms being on the same side of an important double bond. When retinal absorbs a photon of light, a component of this double bond breaks, allowing free rotation about the bond. This change in retinal to an all-trans form triggers a series of alterations in the opsin component of the molecule. The changes in rhodopsin lead to a cascade of events in the cell. The next component in the cascade is a trimeric G protein called transducin, which in its inactive state is bound to GDP. The altered rhodopsin molecule activates transducin, allowing it to exchange its GDP for GTP. The alpha subunit of transducin then activates a phosphodiesterase in the disc membrane. Phosphodiesterase hydrolyzes cyclic GMP, thus lowering the concentration of cyclic GMP throughout the outer segment. As the concentration of cyclic GMP falls, this molecule no longer binds to and holds open ion channels in the surface of the outer segment membrane. Let's compare the action of the ion channels in the light and in the dark. In the dark, the ion channels are open. The influx of the positively charged sodium and calcium ions has a depolarizing effect on the cell. The inward current is opposed by positively charged potassium ions flowing out through potassium channels, a hyperpolarizing influence. The combined result of these ion flows, however, is the depolarization of photoreceptors in the dark. In the dark, the depolarized state of the membrane, which is at approximately minus 40 millivolts, triggers continual transmitter release from the synaptic terminals of the photoreceptor cells. Absorption of light reduces the concentration of cyclic GMP in the outer segment, leading to the closure of the cyclic GMP gated channels. As a result, positive charge carried by potassium ions flows out of the cell more rapidly than positive charge carried by sodium and calcium ions flows in. The cell becomes hyperpolarized, that is, more negative, and decreases its release of transmitter. One of the important features of this cascade is that it provides enormous signal amplification. A single light-activated rhodopsin molecule can activate an estimated 800 transducin molecules. Although each transducin activates only one phosphodiesterase molecule, each phosphodiesterase is capable of breaking down as many as 6 cyclic GMP molecules, resulting in the closure of approximately 200 ion channels. This number of channels represents 2% of the number of channels in each rod that are open in the dark, and their closure causes a net change in the membrane potential of about 1 millivolt. Proteins in the photoreceptor limit the duration of this amplifying cascade and restore the various molecules to their inactivated states. Activated rhodopsin is rapidly phosphorylated by rhodopsin kinase, which permits the protein arrestin to bind to rhodopsin. Bound arrestin blocks the ability of activated rhodopsin to activate transducin, thus effectively truncating the phototransduction cascade. By hydrolyzing GTP, Activated transducin has a built-in timing mechanism to turn itself off shortly after becoming activated. Without activated transducin, phosphodiesterase also turns off. Finally, through a multi-step pathway, the cell converts rhodopsin back to a form that can absorb light. Also in the dark, the enzyme guanylate cyclase again builds up the levels of cyclic GMP. Cyclic GMP opens the ion channels, allowing an influx of ions and depolarizing the cell.
Okay, so that was, I know, a little bit long and probably a little overwhelming if you haven't really spent time looking through the chapter and the rest of these process. But we're going to break it down, but I definitely recommend, we can do this. We are going to come to one for sound transduction a little bit too. So if that was confusing for sight for those, we're going to go break it down some more, but definitely go back and watch that video or other videos that you can find to help you give a visual representation of this process. So dark adaptation. It's a return to sensitivity to low light levels after bright light. The bleached rods must regenerate rhodopsin, and it may take 20 to 30 minutes to see well, so that going back into the dark. In light adaptation, it's a process of adjusting from low light to bright conditions. When you're going out into bright light, your pupils constrict. The cones initially are overstimulated, and it takes about 5 to 10 minutes for them to get fully adjusted to that bright light. All right, initiating the nerve signal. So actually this nerve pathway, that's kind of what the video got into kind of ahead of what we did when it started getting into all that cyclic GMP stuff. So if that's where you got lost, here we go. So in the dark, the rods are depolarized. So they're depolarized at negative 40 millivolts. So I think a lot of times when we think about depolarizing something, we're thinking about going up to 0 or above. But this can still be depolarized at a negative 40 millivolts. So in the dark, when there's no light stimulating it, your rods are depolarized, and cyclic GMP is produced. So cyclic guanosine monophosphate, you can just call it cyclic GMP, is produced. Cyclic GMP is going to bind to the cation channels to keep them open to allow the cell to continuously being depolarized, to allow the photoreceptor cell to continuously being depolarized. Sodium and calcium enter the cell. They call this the dark current because while there's no light stimulation, these are the ions that are flowing in. And the voltage-gated calcium channels in the rod synaptic terminals stay open as well. So as the cell itself is being depolarized, at its synaptic terminals where it synapses with the ganglionic cells, the optic nerve cells, it's going to continuously being releasing its neurotransmitter, which is glutamate. It's continuously releasing glutamate by the rod to the ganglionic nerve to inhibit it. Glutamate hyperpolarizes those bipolar cells, preventing them from exciting ganglion cells. Sorry, I messed up. I forgot the bipolar cells were in there in between. So you have these photoreceptor cells synapsing with those bipolar cells, and as glutamate is released from the photoreceptor cell, it inhibits the bipolar cell. If it inhibits the bipolar cell, it does not send a signal up the ganglionic cell, and there's no image that's produced. We don't perceive an image. And again, this is in dark, so it makes sense. When we're exposed to light, the light hyperpolarizes the rods, right? So the light, as light hits the rhodopsin molecules, it splits the rhodopsin, it converts the retinal to transretinal, and a G protein second messenger system is activated. Your phosphodiesterase, if you remember your enzymes, phosphodiesterase is something that goes in and removes a phosphate molecule. So phosphodiesterase is activated and it breaks down the cyclic GMP. Those cyclic GMP channels are going to close and cutting off that sodium-calcium current so that the cell becomes more negative. You have potassium leaving, you have no cations flowing in, and so you end up with this hyperpolarized cell. So in the dark, our photoreceptors are depolarized. In the light, the cells become hyperpolarized. Once they're hyperpolarized, they stop releasing glutamate. The bipolar cells are no longer inhibited by the glutamate from the photoreceptor cells, so they can release, in turn, glutamate to the ganglionic cells. Now, glutamate is going to inhibit bipolar cells, but it's an excitatory molecule neurotransmitter for ganglionic cells. So ganglionic cells, excitation leads to impulses being sent along its axon to the brain. So remember we said that neurotransmitters are going to function differently depending on the molecules or the receptor cells that they're binding to. So in this case, you have glutamate inhibiting bipolar cells, but exciting the ganglionic cells. Okay. Here is just your book's representation of that. In the dark, you can see the dark current is coming in, the cells are being depolarized, the photoreceptor cell is releasing glutamate to inhibit the bipolar cell. Bipolar cell is inhibited, so there is no action potential generated. In the light, the dark current is cut off, the cell becomes hyperpolarized, it stops releasing glutamate, the photoreceptor cell stops releasing glutamate. This allows the bipolar cell to be activated to release its neurotransmitter glutamate as well, which causes an action potential to be transmitted. So the pathway for that, you have your photoreceptors, your bipolar cells, and your ganglionic cells. The ganglionic cells, axons bundle at disc to form the optic nerve. Okay, so you have at the back of your eye, all of the photoreceptor cells, their axons starting to gather to form your optic nerve that leads back into your brain. The optic nerve exits the back of the eye and converge at the optic chiasm. The medial axons cross to the opposite sides of the brain, whereas the lateral axons remain on the same side. So for part of your optic pathway, the nerves coming from your left eye will be transmitted to the right side of the brain, whereas the right will be transmitted to the left, and then you have some that stay on the same sides. Your optic tracts are the ganglion cell axons from both eyes. Most axons go to the lateral geniculate nucleus of the thalamus, and thalamic neurons axons project to the visual cortex of the occipital lobe. That's the ultimate destination for the optic pathway. So the left and right eyes have overlapping visual fields. This allows for stereoscopic vision, which is our depth perception. So the fact that we have this kind of overlapping of our left and right allows us to see things at different depth, and we can focus on things at different depth. You have some optic tracts projecting into the midbrain. The superior colliculi coordinate reflexive eye movement, so if you see something coming out of the corner of your eye, it'll jump over there. And the pretectal nuclei coordinate pupillary reflex and lens accommodation. So here you go, you have your right eye depicting, so this is the inferior view, so we're under the brain if that was confusing for you. You have your right eye detecting, it's monocular vision, left eye for monocular vision, and then together they create binocular vision. So our brain normally will, you can close one eye and still kind of get an image. If we have both eyes open, our brain will kind of blend those two images and give us this binocular vision. It's not true for all mammals. Horses, because their eyes are so far set, they have monocular vision and binocular vision like we do, except that their brain can interpret things from one eye. So they can, what's on their left side, they can see that. They have the image on the right side, and then if it's right in front of them, it's far enough in front of them, they can see it monocularly. But typically they have binocular vision. That's why when you go around horses, typically when you go from one side to the next, especially if you move behind them, their brain has a little bit of a lag. So while their left eye might see you and sense you, as soon as you cross behind them and cross to their right side, there's a lag, just milliseconds in their brain from, oh yes, there is, that person is supposed to be there. So when all of a sudden you pop up out of nowhere from their vision on their right side, it kind of spooks them, which is one of the reasons why horses can seem so skittish. It's because their eyes don't always see, or they're not always talking to each other. Okay, you don't care. You're not here for horse physiology. All right. Okay, and so that was basically our eye. That was vision. If you have questions, go back through and read that again. Email me for sure. All right, now getting into sound. So ear structure. The ear detects sound and head movement. The signals are transmitted via the vestibular cochlear nerve, that cranial nerve 8. Your external ear, there are 3 parts of the ear. The external ear has your auricle, which protects the ear entryway and directs sound waves inward. It's this outer portion. The external acoustic meatus, the ear canal, extends to your tympanic membrane, and it also has earwax. That's where earwax is produced. The tympanic membrane is your eardrum. It vibrates when sound waves hit it, and it separates the external and middle ear. So 3 structures of the external ear. Here is just a lovely little breakdown of all of those portions. Middle ear. The middle ear contains the air-filled tympanic cavity. So again, we're talking about from the inside of the tympanic membrane up to the inner ear. It has this bony wall that separates it from the inner ear that has 2 membrane-covered openings, the oval window and the round window. So your middle ear is essentially from your tympanic membrane to this bony wall separating it from the inner ear. Okay? It has the auditory tube, the eustachian tube, which is a passage extending from the middle ear to the nasopharynx in your upper throat. Your middle ear infections often result from infections spreading from the throat up through the auditory tube. You may notice that's kind of when you pop your ears, you yawn, or you're on the plane, you kind of plug your nose and you push air out. It's going through this eustachian tube, this auditory tube. The middle ear also has your auditory ossicles, which are 3 tiny bones, your smallest bones in your body. The malleus, incus, and the stapes. And they are all there to transmit signals, vibratory signals from the tympanic membrane to the inner ear. The ossicles amplify sound waves, so they not only transmit them, but they also amplify those sound waves, and they vibrate along with the eardrums. The stapes moves in and out of the oval window, initiating pressure waves in the inner ear fluid. And 2 small muscles restrict ossicle movement during loud sounds. So just to make sure that the little ossicles don't vibrate too far, don't vibrate too hard rather, you have these muscles, the tensor tympani and the stapedius, that are going to restrict their movement just ever so slightly. Here is a representation of that. You have your tympanic membrane, you can see your malleus, your incus, and then your stapes up against this oval window. And again, they'll kind of transmit the signal from the tympanic membrane through the stapes and the oval window into the inner ear to transmit those vibrations, amplify and transmit. So your inner ear is the spaces within the petrous part of the temporal bone. So inside of the temporal bone, it has 2 different parts, basically. It has a bony part and a membranous part. It has this bony labyrinth and the membranous labyrinth. The bony labyrinth is exactly what it sounds like. It's the bone portion. It has perilymph, which is its interstitial fluid. And then inside of the bony labyrinth is this membranous labyrinth, which are a series of tubes that are filled, fluid-filled tubes within the bony labyrinth itself. And it contains endolymph. It also has structures for hearing and equilibrium. Sorry, my dog. The inner ear has 3 main regions, the cochlea, the vestibule, and the semicircular canal. We'll see that the cochlea is very important for hearing. The semicircular canal is more so for equilibrium. The cochlea has the membranous cochlear duct. The vestibule has 2 parts, the utricle and the saccule. And the semicircular canal contains the semicircular ducts, which have fluid in them. Okay, so here you go. You have your cochlea, your vestibule, which is this middle part, and then the semicircular canals. You can see that this outer portion is the bony labyrinth. So it is this entire structure is this bony labyrinth. And then inside of this bony labyrinth is this pink membranous labyrinth, the tube-filled system, or the fluid-filled tubule system running all through the inner ear. This is your cochlear branch of the vestibular cochlear nerve that's going to be sending hearing signals, sound signals. And these are just cross sections of these tubes. We'll look at those a little bit more. Okay. So, structure for hearing. Your cochlea, like I said, is very important for hearing. It's the snail-shaped chamber of the inner ear, that really wrapped around one. It has a motolius, which is the bony axis of the spiral, a cochlear duct, which is the membranous labyrinth within the cochlea. Remember, so the outer portion is the bony one, and then it has those tubes with the fluid in them, the endolymph in them, which is the membranous labyrinth. It has a roof, which is the vestibular membrane, and the floor, which is the basilar membrane. And then it has also the scala vestibuli and the scala tympani. These are both chambers and kind of sandwiched between these membranes. And this sounds confusing, but it'll make sense when we see the cross section of it. Also within the cochlea is the spiral organ, which is a sensory structure for hearing. You might also hear this called the organ of Corti. This is within that cochlear duct. It's a thick sensory epithelium. So it's a thick sensory epithelium consisting of hair cells and supporting cells on the basilar membrane. So we'll see that hair cells play a very important role in hearing and equilibrium. Your hair cells are receptors that release neurotransmitter to the sensory neurons. And the base of the hair cells synapse with those sensory neurons in the ganglia of the motolius. Okay, so you have your cochlea, which is this spiral organ, and it starts down here, and it wraps around, and as it spirals, it's going up. So each of these little cross sections is just a cut from this spiral. So you can see that you have your scala vestibuli, this big chamber up here. You have your cochlear duct, this chamber, and then you have your scala tympani, this chamber. Okay, this membrane right here. This one is your vestibular membrane, and then you have your basilar membrane. Okay? So the vestibular membrane is underneath the vestibuli. and the basilar membrane is above the tympani. So they detect sound. So again, as the hair cells within this fluid are stimulated, sound waves will travel up this spiral, and they'll stimulate hair cells. And those hair cells transmit their signal from these fluid-filled chambers. It vibrates. They release their neurotransmitter, and then it travels down this cochlear nerve. Within the cochlea, we have the spiral organ. So you have your vestibula, you have your tympani, these chambers, your cochlear duct, and then within this cochlear duct, you have the spiral organ that actually has, I think we have, oh, I thought we have one more. Can I? No. And you might be able to barely see it. There's little hair cells that are projecting up here. And then at the base of these cells, they're synapsing with this nerve that will then take it to the cochlear nerve. So the hair cells are moved, they release their neurotransmitter that travels into the brain. Okay, so looking at hearing. The pathway from sound wave to nerve signal. Sound waves vibrate the tympanic membrane in the outer ear. The ossicles vibrate in the middle ear and transmits the waves to the oval window. Fluid-filled pressure waves in the scala vestibulae and push the vestibular membrane, causing pressure waves in the endolymph of the cochlear duct. So this is in our inner ear. So we go from outer, middle, now we're in our inner ear. The sound waves go from being just vibrations in the air to now they're vibrating this fluid, this endolymph of the cochlear duct. Specific regions of the basilar membrane will move depending on the sound wave frequency, whether it's a higher pitch or lower pitch. This movement of the basilar membrane will distort certain hair cells, causing changes in their neurotransmitter release. The sensory neurons within the cranial nerve 8, the vestibulocochlear nerve, are stimulated to fire based on this neurotransmitter release. And the pressure transmitted to the scala tympani is absorbed by the round window. So this is just kind of a tacked on the end there. But you remember that the middle ear has both an oval window and a round window. The oval window is what the stapes attaches to, and it's what it vibrates against to send the sound wave signal. The round window, because the cochlear duct is enclosed within a bony capsule, as the pressure waves increase from the sound waves, that has to be relieved somewhere, and so it's absorbed by the round window. The round window is a membrane that bulges out. So as the stapes is kind of beating on the oval window, creating a sound pattern, the round window is right below it, bulging out to absorb that excess pressure from the fluid is receiving. Okay, so you have the external acoustic meatus, so your ear canal receives the signal, goes from the tympanic membrane, travels through the ossicles into the scala vestibuli. So they kind of uncoiled the cochlea here. You know, normally it would be very much curled up. They kind of stretched out a little bit. So the sound wave travels through the cochlear duct and across into those hair cells, into that spiral organ, which is then absorbed by the nerve branch. So stimulating the cochlear hair cells. The inner hair cells contain ion channels at their tips and a tip link proteins that connect them. So each hair cell is connected to the next one by a tip link protein, and they have ion channels at those tips. The hair cells are bathed in potassium-rich endolymph that is far more positive than the fluid inside of the cells. So the fluid, the endolymph within the cochlear duct that the hair cells are in, it's got a lot of potassium. So as soon as those ion channels open, the potassium can move in. When the basilar membrane moves up, the hair cells are pushed into the tectorial membrane and their tips are tilted, pulling tip links. So remember, this is within that spiral organ. It has part of it is the tectorial membrane. So your basilar membrane is moving up and it's pushed into the tectorial membrane, which then causes, if I push my hair down, the hair cells get distorted. As they're distorted, they're opened up to release, to allow potassium to diffuse into the hair cell and depolarize it. Once the cell is depolarized, it releases more neurotransmitter from its base, exciting the sensory neuron, which can then fire action potentials. When the basilar membrane moves down, the process quickly reverses, and this can occur 20,000 times per second, which is just incredible to me that the basilar membrane can move up and down and reset itself, transmitting signals 20,000 times per second. It's very impressive. Okay, so here are these hair cells. They're within this endolymph, this nice potassium-rich endolymph. As this basilar membrane starts to move up, it pushes these hair cells up against the tectorial membrane, and they'll start to move. And as they move, these tip link proteins will kind of make sure they all shift together, and that will also open. You can see there, it's opening up these channels to allow potassium to rush in. As potassium rushes into the cell, it'll allow it to depolarize. Once it's depolarized, it'll send its signal out through the cranial nerve 8, that vestibulocochlear nerve. Like I said, I have a video for this too. All right. I think we missed it. No, okay. So sound, perception of sound. You know what? I'm going to go ahead and watch this video before we get into this. All right.
Responsible for translating variations in air pressure.
Sorry, I was watching it on a little fast speed. And yes, I was watching Gourmet Makes as well.
From speech, music, or other sources into the neural activity necessary for our perception and interpretation of sound. The ear can be divided into 3 principal sections: the outer ear, the middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear, or pinna, and a canal that leads to the eardrum. The configuration of the external ear amplifies sound, particularly at the frequency ranges of 2000 to 5000 Hz. Not coincidentally, most human speech sounds are distributed in the frequency range around 3000 Hz. From the ear canal, the sound waves vibrate the eardrum, which in turn vibrates 3 tiny bones in the middle ear: the malleus, incus, and stapes. The stapes vibrates a small membrane at the base of the cochlea, the oval window, which transmits amplified vibrational energy to the fluids of the cochlea. The round window serves to dampen the movement of fluid within the cochlea. The complex structures of the inner ear convert sound into neural activity. In mammals, the auditory portion of the inner ear is a coiled structure called the cochlea. The region nearest the oval window membrane is the base of the spiral. The other end, or top, is referred to as the apex. Along the length of the cochlea are 3 parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The principal elements for converting sounds into neural activity are found on the basilar membrane, a flexible structure that separates the tympanic canal from the middle canal. Let's take a closer look at the basilar membrane by unrolling the cochlea and peering inside. The basilar membrane is about 5 times wider at the apex of the cochlea than at the base, even though the cochlea itself narrows toward its apex. It vibrates in response to sound transmitted to the fluid-filled cochlea by deflections of the oval window initiated by the bones of the middle ear. High-frequency sounds displace the narrow, stiff base of the basilar membrane more than they displace the wider, more flexible apex. Mid-frequency sound maximally displace the middle of the basilar membrane. Finally, lower frequency sounds maximally displace the apex. Within the middle canal and atop the basilar membrane is the organ of Corti, the collective term for all the elements involved in the transduction of sounds. The organ of Corti includes 3 main structures: the sensory cells, or hair cells, an elaborate framework of supporting cells, and the terminations of the auditory nerve fibers. The heights of the stereocilia increase progressively across the hair cell, so the tops approximate an inclined plane. Atop the organ of Corti is the tectorial membrane. The stereocilia of the outer hair cells extend into the indentations in the bottom of the tectorial membrane. The movements of fluid in the cochlea produce vibrations of the basilar membrane. These vibrations bend the stereocilia inserted into the tectorial membrane. Depending on the direction of the bend, the hair cells will either increase or decrease the firing rate of auditory nerve fibers.
Okay. So basically what it's saying is that that organ, of course, you remember, is just the spiral organ. It's the primary organ for hearing. So as the basilar membrane moves, it shifts the hair cells, and they can fire and send their nerve signals more. All right, so how then do we still perceive that sound? So perception of sound is done by the pressure waves established from vibrating objects. The vibration pushes molecules, which transfer the energy from one molecule to the next. So again, it's just kind of just moving the energy of vibration in the air into vibration of the fluids, which then turns it into this electrochemical signal. All right, different things that we can detect. Pitch depends on the frequency of the vibrating object, and frequency is the rate of vibration. So something that vibrates higher frequency has a higher pitch, lower frequency has a lower pitch. Humans can hear 20 to 20,000 megahertz, or hertz rather, and variations in pitch are detectable due to variations in the stiffness of the basilar membrane from the oval window to the cochlear apex. Like the book said, closer to the base of the basilar membrane, the basilar membrane closer to the base of the cochlea is going to be stiffer. As you go more up towards the apex, it becomes more flexible. So high frequency sounds excite the stiff cells in the basilar membrane near the oval window, and low frequency sounds are going to excite near the apex. Yeah, there's that link to that video. So again, you have these high frequency sounds very close to the oval window being detected here, whereas as you go up towards the apex, that's when you're going to start to detect your more lower frequency sounds. Loudness depends on wave amplitude, the degree of molecular compression. So louder sounds create larger movements of the basilar membrane. So degree of molecular compression, how much are those sound waves? How much is are the molecules being pushed and vibrated and moved through that sound? I have a friend who cannot control her pitch. That is just something that, as a child she went to the doctor for, and she just has no ability. I'm not sure why, but apparently it's been going on since she was a kid, so it's legit. So she can't control her pitch. So to convey any kind of excitement, she can't get higher and clear. She can't, really go low and say I'm depressed. So she just is loud. So when she's excited about something, she just gets louder. When she gets angry about something, it's hard for her to really, kind of convey that anger. So she just gets loud. When she's sad, she just gets quiet. She has no other way of kind of controlling emotion through her voice other than just being loud. It's nice being her friend sometimes. All right, so larger movements cause faster rate of nerve signals and a larger number of stimulated cells. So essentially, the louder something is, the more stimulation it's going to cause, the more displacement of air cells, the more displacement of hair cells that it will cause. That then in turn sends a larger nerve signal. The temporal lobe's auditory cortex interprets that as loudness. Those excess signals, or those the high rate of signals, is interpreted as loudness. That's measured in decibels. 0 decibels is a threshold for hearing. Maybe you can hear my dog scratching right next to me. And the energy of sound increases 10 times for every 10 decibel increase. So for every decibel increase of 10, you have to have a 10-time increase of energy. Sorry, she's going for something. Okay, the auditory pathways. Those stimulated hair cells in the cochlea initiate the nerve signals in cranial nerve 8, extending into the cochlear nucleus of the medulla oblongata, part of your brainstem. The secondary neurons transmit signals to the inferior colliculus of the midbrain or to the superior olivary nucleus, 2 parts of the midbrain. So the inferior colliculus coordinates head-orienting reflexes to sounds. So, I heard something, let me turn real quickly. And the superior olive localizes sound and initiates reflexive contraction of middle ear muscles to help make sure that they're not amplifying too many loud sounds. Your MGN is in your thalamus, your medial geniculate nucleus, I think. filters signals and relays them to the primary auditory cortex and the temporal lobe for conscious perception of that sound. So you have a couple different places it's going, the medulla oblongata, some portions in the midbrain, and then the temporal lobe for conscious perception, the primary auditory cortex. Here's just kind of what that looks like if you enjoy having kind of the graphic representation of that. Okay, now getting into equilibrium and head movement. Equilibrium is an awareness and monitoring of head position. It's monitored by the vestibular apparatus, which is the utricle and the saccule, and the semicircular ducts. So there's other two portions of the bony labyrinth within the inner ear. Information is sent to the brain to help keep our balance. Vision and proprioception also help to our proprioceptors in our joints, and then that visual pathway. The utricle and the saccule detect static equilibrium and linear acceleration, while the semicircular ducts detect angular acceleration. Within the utricle and the saccule, they have these things called the macula. The macula is the receptor for static and linear acceleration. It's within the vestibule. It's composed of a layer of hair cells and supporting cells, so more hair cells here. The hair cells have stereocilia, which are shorter hairs, and then one long kinocilium. These hairs project into what's called an otolithic membrane that is filled with otoliths, which are stones. Lith is it means stone. If you ever have like what is it, lithotripsy, when you have kidney stones or gallstones lasered away? Lith, sorry, is stone. So head tilt shifts otolithic membrane and bends those stereocilia. So as you kind of move your head and tilt it yes and no and shake it yes and no, that's going to then shift that otolithic gelatinous fluid, this otolithic membrane, which in turn will rock those otolithic crystals that are going to bend and disrupt the stereocilia. Bending the stereocilia toward that one long kinocilium depolarizes the hair cells and increases their transmitter release. This increases impulse frequency in the vestibular part of the cranial nerve 8, and the opposite direction occurs if bending is away from the kinocilium. So as we kind of move our head and we move all around, the stereocilium are going to get bent and shifted, and if they get bent towards the kinocilium, they're going to depolarize, and when that particular hair cell, when that particular nerve fires, it's going to send a signal saying, hey, now I'm active, so the head position is this way. And the combination of all of those hair cells sending their signals either depolarizing or not depolarizing will help give our brain kind of an idea about, okay, where is our general position in space? Again, if they bend away from the kinocilium, they hyperpolarize. So you had your hearing structure, your cochlea over here, and then for our equilibrium structures, we have the vestibule and then the semicircular canals. Remember, within your vestibule, you have your saccule and your utricle, and then the receptors for equilibrium are the macula. So you have your utricular macula and your saccular macula. There's some tricky words in this lecture for me. And then the semicircular ducts, notice that they are oriented anteriorly, posterior, and then horizontal. So they're kind of able to detect all of those angular movements. Right here is the otolithic membrane. So it has the gelatinous layer filled with these otoliths, with these crystal or little rocks rather. You have your one tall kinocilium, your smaller stereocilia. You see they're kind of angled here, so they can kind of pull and shift with this gel membrane. Into the macula portion of it, you have your supporting cells surrounding the hair cells, and then these the vestibular branch of the vestibulocochlear nerve wrapped around those receptor cells, all gathering together. So in the upright head position, the stereocilia are parallel to the kinocilium. The neurotransmitter is released at a regular interval. A steady rate of nerve signals are transmitted along the vestibular branch of the cranial nerve 8. So you have this kind of standard nerve frequency as long as your head is upright. There's really no increase or decrease in firing. As you start to lift and tilt your chin upward, you have the stereocilia bent toward the kinocilium, and you have this excitation. You have this increase in nerve fibers. As you tilt it downward, the stereocilia bend away from the kinocilium, and that's going to cause inhibition. For the angular acceleration, the base of each semicircular canal has a region called an ampulla, which is just a kind of a swollen portion. It contains the crista ampullaris with hair cells and supporting cells. There are stereocilia and kinocilia of hair cells embedded within the gelatinous cupula. And when the head rotates, the endolymph pushes against the cupula. So as your head is turning and rotating, the endolymph pushes up against the cupula. The cupula bends the stereocilia and changes the hair cell voltage, so it polarizes or depolarizes them. If they're bent toward the conocilium, the hair cell depolarizes away, it hyperpolarizes. Same thing as with in the vestibule. The neurotransmitter release from hair cell changes as they're depolarized or hyperpolarized, and the firing rate changes then to the cranial nerve 8. So here within the semicircular ducts, you see you have this kind of crista ampullaris structure, this portion right in here. There are your stereocilia and that long kinocilium. This is that cupula with its endolymph. I'm sorry, here's your endolymph. The cupula has the gelatinous membrane. So with your head still, no big deal. As your head starts to rotate, that shifts the endolymph. The endolymph then pushes this cupula filled with that gelatinous membrane, that otolithic membrane, and that is going to bend the stereocilia. So the pathways for equilibrium. The signals from the macula or crista ampullaris, so the macula and the vestibule, the crista ampullaris and the semicircular canals are conveyed by the vestibular branch of the cranial nerve 8. Those axons terminate in the vestibular nuclei or the cerebellum. So you go from the vestibular branch of the nerve into the vestibular nuclei or the cerebellum. And the vestibular nuclei, they help control reflexive eye movements and balance. The cerebellum helps coordinate balance and muscle tone. Remember the cerebellum, that's kind of one of its main functions, is just kind of coordinating conscious muscle movement with the rest of our body and perceived movements, fine-tuning things and acting as an error corrector. So if all of a sudden our equilibrium is saying, hey, your equilibrium is off, our body will kind of catch us. Hopefully. Or if your equilibrium is really off, you're walking normal in a straight line, and then all of a sudden you just kind of trip and fall, and you feel really dizzy because there's potentially something, you just have like a misfiring in your vestibular cochlear nerve. The vestibular nuclei and cerebellum both send those signals to the thalamus, and the thalamus relays information to the cerebral cortex for awareness of body position. So it's how we can kind of know, it kind of goes this indirect pathway from the vestibular branch into the cerebellum, and then up into the thalamus, finally to the cerebral cortex, so we can actually know where we are. And that is it. That is all for our lectures.